2018, a rogue scientist announced genetically modified embryos to a room full of shocked scientists. This was particularly crazy because at the previous conference they said, "Don't do this." But the scientist was too much of a bad boy and said YOLO. Scientists responded by being big mad, universally condemning his work. He was fired from his job at his university and he was sent to jail for 3 years. But now he has been released from jail and it turns out that the people love him. He refuses to apologize for his actions and he even took time to flex on his haters. But this bad boy can't help himself and he's back in the lab working on what got him sent to jail. And other scientists are starting to acknowledge that embryonic gene editing might be the future. This is the He Jiankui Affair. He Jiankui, who I'll call JK, wasn't always a bad boy. He received his PhD at Rice University before doing his postdoctoral work at Stanford, where he worked with CRISPR-Cas9, a useful tool for making genetic modifications. He then returned to China where he was given a laboratory and a relatively large amount of money. Being a scientist in China is very difficult, and for most scientists, receiving a lab and money would have satisfied their desires. But JK wasn't most scientists, and he was going to do something that no scientist had ever done. Genetic modification isn't new. Humans have been breeding plants and animals for thousands of years to get desired traits. This is why your vegetable aisle came from the mustard plant. It's why your ancestors wouldn't recognize our corn. And it's why we have so many types of dogs. That's right, your pug dog was somehow the result of breeding for desired traits. But the discovery of CRISPR-Cas9 allows for the precision editing of our DNA. We can now give ourselves our desired traits. In other words, we can spell check nature. Somatic gene editing works by manipulating your DNA after you've been born, at which point you become a genetically modified organism. Scientists can turn you into a mutant with superpowers. Scientists became excited at the prospect of curing all of the diseases. But there's a catch. The human body does not want to become a bioreactor. If it was easy to modify your DNA, every virus would immediately use your body as a factory to mass produce itself. There's over 30 trillion cells in your body and it's incredibly difficult to modify all of them in a way that's permanent. This is where germline editing comes in. What if we just modify the genes of the embryo? This would even allow the genetic modification to propagate into your children. It's much easier to modify you when you only exist as one cell, and then let nature take its course and let one cell become trillions of cells. This would allow us to eradicate human diseases off the face of the planet for the rest of human existence. We don't know the limits of what is possible with this scientific technology. For JK, the solution was obvious. Somatic gene editing was too difficult and expensive, so he got 2 human embryos and modified their CCR5 receptors. The CCR5 receptor is what allows HIV to enter the cell. His gene edit would be a vaccine for AIDS. By now you're probably saying, why would he do this? These babies were not at risk for HIV. Most scientists would start with an uncurable disease that had a 100% chance of death because it's considered less morally unacceptable to kill someone while trying to save them. Modifying the CCR5 receptor didn't appear to have any immediate benefits and could have a chance of significant harm. JK announced the birth of healthy, genetically modified twins. But this claim was fact-checked by true Chinese patriots. One of the problems with CRISPR-Cas9 is that it can modify parts of your DNA that you do not want it to. In fact, this may have actually happened, and recent evidence suggests that these off-target effects might be worse in human embryos. There's also the problem of genetic mosaicism, which means that the genetic modification didn't propagate to all cells equally. This is the reason your female calico cat is different colors, because the DNA expressing color is different in different cells, which means that the children might not even be protected from HIV. And to top it off, he didn't have valid informed consent, which means that the experiments were never ethical to begin with. All of this evidence resulted in JK being arrested. But embryo editing isn't crazy. For example, scientists recently modified the embryos of chickens to prevent bird flu. And JK is right, somatic gene editing is difficult and expensive. A recent somatic gene therapy was approved for sickle cell disease and it costs over $2 million. Doing the same thing in the embryo might cost 1% of that, and it would automatically ensure that your children don't die of sickle cell disease. The pressure to erase the biggest killers on the planet is enormous, which is why even before JK did his thing, scientists were saying we should probably try this. JK's biggest mistake was that he was a bad boy who rushed things too quickly. But we are now looking at a future where your children's traits might be market-based and consumer-driven, and you might be able to purchase the eradication of Alzheimer's from your child in the same way that you would purchase an iPhone. Do you have any ideas for videos I should do in the future? Let me know in the comments. And remember to like and subscribe to support me.